Now we are going to discuss about a quantitative, autosomal, recessive, genetic human disorder. Now what that is? Too many terms, right? It is thalassemia. This thalassemia is a type of anemic condition. So you are going to find the patient would be anemic, or I would say anemia is what you are going to deal with. Now because it is a human genetic disorder, either it would be mutational in nature or it could be deletion of the allele which is responsible for production of alpha and beta globin chains of hemoglobin molecule, not hemoglobin, right? What am I talking about? Firstly, as I told you, it is a quantitative, it is an autosomal, it is a recessive disorder of blood, okay? And blood, that too, in it affects the hemoglobin molecule. Now hemoglobin has 4 chains, 2 alpha, 2 beta. So this hemoglobin will have these alpha and beta globin molecules altered, or it will be produced in lesser quantity. Now what happens is, you have to remember, this thalassemia is of 2 types, alpha and beta. In alpha we are dealing with chromosome number 16, and in beta we are dealing with chromosome number 11. Now an individual acquires one chromosome from mother and other from parent. Because this is a recessive disorder, if both the parents are carriers, then only the offsprings are going to have the disorder, alright. Supposedly we have this as the carrier mother and this as the carrier father, then only there is a probability of having an individual or an offspring who is suffering from the disease, okay, because it is a recessive autosomal disorder. Moreover, it is quantitative disorder. That means due to the absence of this diseased recessive gene, there would be decline in production of something, okay. The protein that these particular genes are coding or alleles are coding, they would be produced in lesser quantities. So that is why this is a quantitative disorder. And what it produces, in one case it produces alpha globin chain and in other case it produces beta globin chain. How does it occur? Either there is going to be mutation, either it is going to be the mutation which will be a cause, or it could be deletion of chromosome itself, okay. Mutation or deletion could be the reason. Now if it happens that in chromosome 16 there are 2 HbA1 gene and 2 HbA2 genes on chromosome 16, 2 of them come from mother, 2 of them come from father. Supposedly mutation or deletion occurs in these of the genes on chromosome number 16. The patient suffering from thalassemia will, out of the 4 I am talking, out of the 4, if the mutation or deletion has, then the patient who is suffering from thalassemia will have lesser formation of alpha globin chain in the hemoglobin molecules. That is why it is quantitative. Similarly, in the case of beta type thalassemia, at chromosome number 11 we have the gene HBB, okay. This is having 2 genes, one from mother, one from father. Earlier it was 4. Now out of these 2, if someone is, if one is mutated or deleted, then what will happen? Now the beta globin chains formed will be lesser in number, and hence the quantitative disorder of thalassemia will have lesser hemoglobin molecules, or the hemoglobin molecules which would be formed, they will be having lesser number of beta globin chain, and once the chain is not present, lesser is the chain, lesser would be the globin molecules, certainly the protein will not be formed, and because of that the hemoglobin molecules will decline in number. That will lead to the condition known as anemia. Alright, so this is what you have to keep in mind about thalassemia. Certainly it is different from sickle cell anemia, because sickle cell anemia was qualitative. If you remember, when we did mutations over there, we had taken the classic example of sickle cell anemia. Now that was qualitative. Why it was qualitative? Because a specific thing was totally absent and it was replaced by something. When it was replaced by something, it led to a condition where hemoglobin could not be formed. Over here, the quantity being lesser hemoglobin is being formed and it is leading to anemia. That is why thalassemia and sickle cell anemia are totally different types. Okay. Sickle cell anemia was a type of point mutation where the codon which is responsible for coding for glutamic acid was replaced by valine, and altogether the quality changed. Over here, it is not the quality which is changing. What is changing is that the genes are altered, and because of that the production is not taking place. Because the production of alpha and beta globin chains is not taking place, we do not have much of hemoglobin, and that leads to anemia, and this is what thalassemia is all about. Also, you have to keep one thing in mind, that for the diseased condition to occur, either the parent should be, no, not either, both the parents should be heterozygously diseased. Okay, the parents should be carriers, then only the offspring will be diseased. In other case, we can have one homozygous dominant parent and one heterozygous, homozygous recessive and one heterozygous, then also the individual could be diseased. But classic way of showing it is that both the parents are carriers and the offspring is thalassemic. So this is all you have to keep in mind when we talk about thalassemia as an autosomal recessive genetic disorder found in